Hello, our topics today for video notes is DNA replication, which is from chapter 12.3, and RNA in chapter 13.1. So here's your essential question. Can you summarize the events of DNA replication? So moving right into chapter 12.3, we're looking at DNA replication. Think back. What does this word replicate mean? You got it right. It means we are going to copy our DNA. And think about, when does this happen in the cell cycle? So replication is going to happen during our S phase, and we have to copy the DNA so we can provide the new cell that's created a same complete set of DNA. I kind of think of it as like an instruction manual so it can function. So let's look at the details of how DNA replication works. So first of all, DNA must unzip. So we are going to copy this middle section of DNA here. So we must unzip it. And when we unzip it, the next thing that occurs is that there are enzymes that help with this process. So if you look up here, the blue spheres represent DNA polymerase, and we see ASE enzyme, which means it's an enzyme. The enzymes attach to the single strands of unzipped DNA. This DNA polymerase is going to work from the top down, as you can see by the arrow, and this other polymerase worked from the bottom of that separated strand up. And as they attach the single strand of DNA, they're going to add one nucleotide at a time to the new strand. So let's zoom in on this blue sphere right here, that DNA polymerase. And right now it's adding a red piece on, which is a guanine. So the guanine is going to add to its complementary nucleotide with the complementary base pair, which would be G's match with C, so guanine and cytosine. And so the DNA will be replicated by the DNA polymerase until that section is complete. Now, the tops and the bottoms of DNA strands are really challenging to replicate. So there's a different enzyme that helps with that process, which is telomerase. And remember, ASE, it's an enzyme to help speed up that process. So what happens at the top and the bottom of DNA, which we call telomeres, these enzymes will work at those sections that are very hard to replicate to make sure that they add the individual nucleotides at those places.
All right, so there are differences between DNA replication in prokaryotes and in eukaryotes. And so in prokaryotes, remember, think back to our unit in cells when we talked about prokaryotes. Bacteria are examples where they have circular DNA, one long circular strand that connected. And so what happens is they basically just unzip at one side, and they just zip, zip, all the way around until they get to the end of the circle. So that's prokaryotes. And in eukaryotes, the situation with them is their DNA is 100 times larger, and it's linear. And so it's just a long strand. And so what happens is the DNA polymerase has to attach at multiple places within the long strand of DNA so that it can replicate the whole amount correctly with all those little nitrogen bases and letters. They have to match up. So there's multiple places where the polymerase will attach to the long DNA strand in eukaryotes. All right, so now we're moving on to chapter 13, section 1, talking about taking our DNA, our instructional manual, and using it to make something very important to our bodies, which is called proteins. And so what happens is we need to decode DNA, and we use something called RNA to help us do this. So RNA stands for ribonucleic acid, and it is a very important molecule necessary for a protein to be made, hence why this section is called protein synthesis, to put together a protein. So there are similarities and differences to RNA and DNA, and you need to know them. So I think once I finish talking about them, you should fill this data table out. So we have DNA on the left, RNA on the right. And so you notice DNA is deoxyribose, and RNA is ribose. And so they both have sugars, but they have actually a different kind of sugar. So DNA has deoxyribose, hence the os, root word sugar, and RNA has just ribose. When you look at their nucleic acids, DNA has the adenine, the cytosine, the guanine, and thymine. But when you look at RNA, it also has adenine, cytosine, guanine, but instead of the thymine, it now has this new nitrogen base called uracil. And finally, the last difference is that the DNA is actually double-stranded helix shape, and the RNA is actually a single strand. And when we get more into the steps of the process of protein synthesis, we'll get more into the single-stranded piece.
All right, so we are learning about this molecule called RNA, ribonucleic acid, and RNA's main function is protein synthesis. So there are 3 different types of RNA, and with each different type, they have different names and different roles that they play in protein synthesis. So the first one's called mRNA, and we call it mRNA for messenger RNA. And the reason why we call it messenger RNA is because it's going to carry the instructions from the DNA out of the nucleus and into the cytoplasm for the next step of protein synthesis. The next type is called rRNA, short for ribosomal RNA, and this is where the proteins are assembled. And then we have tRNA, or transfer RNA, and transfer RNA carries the amino acids to the ribosome to match with the mRNA strand. So 3 different types of RNA with 3 different roles that they play in this whole process of protein synthesis. So here are the questions to fill out on the Google form. First question is, what is the name of the enzyme that helps replicate DNA? And what are the 3 differences between DNA and RNA? Okay, so make sure you fill out those questions and see you tomorrow.